This is a GCSE presentation on communicable diseases. To get started, all you have to do is try and think up as many words as possible that you can connect to the word disease. Should it be specific examples of them, methods in which they transfer, or actions we take to prevent the transfer of diseases. Pause the video, make up as many words as you can, and then come back with gather your thoughts. We'll go through the rest of the presentation. So we're going to look at a lot of different things during this presentation. We're going to look at how pathogens are categorized. We're going to look at how they can spread, and we're going to make some comparisons on how we prevent them from spreading from one organism to another. So in the exam, there's a number of very short, quick questions that I can ask you for between 1 and 2 marks linked in with this topic, and it's straight up, you need to memorize the answer to each of these 4 questions. So first off, what is a pathogen? Well, a pathogen is a disease-causing microorganism. You have to talk about microorganisms, and you have to talk about the fact that it causes disease. There are plenty of microorganisms that do not cause disease. They would not categorize as pathogens. So please make sure you mention both disease-causing and the word microorganisms. There are 4 main types: viruses, bacteria. You should be aware of definitely fungi. You may have known fungi but not known it's classified as a pathogen, and protists. We'll come back to them in a little more detail later on. What is a communicable disease? Well, communicable disease is simply a disease that can spread, but in the exam you have to say that it is an infectious disease, i.e. it can spread, and that it's caused by pathogens. So that's often a 2-mark question. You get 1 for saying it's an infectious disease and 1 for saying it's caused by pathogens. And the last question, malaria is spread from person to person through mosquitoes. Mosquitoes are a vector. What is a vector? Well, a vector is a living or non-living factor. In the case of malaria, it's a living one with the mosquito, but they can be non-living as well. So it's a living or non-living factor that transfers a pathogen from one organism to another. So there's 3 parts to that explanation. What a vector is, you need to learn that definition off by heart. So you need to know some of the properties of each of the different types of pathogen. You need to be aware of the relative size, so you don't know the exact numbers, but you need to be able to say that the viruses are always the smallest, followed by the bacteria, and the largest will be a fungi or a protist. In terms of how it causes illness, viruses actually get into a host cell and destroy the cell, and as they destroy the cell, lots and lots of new viruses that have been made inside that cell are released out and infect more cells inside an organism. Bacteria reproduce very, very rapidly, and because all bacteria release toxins or poisons, they basically can produce large volumes of toxins in a very short space of time, which obviously has a detrimental effect on the organism that's infected. In terms of fungi, fungi often feed parasitically, so they will feed off dead decaying matter. And we've got our protists, which again, they can cause disease directly, or they will also release toxins. Normally in the exam, when they talk about releasing toxins, though, they are talking about bacteria. And there are some specific examples of diseases that you need to know by name, and you need to know some of the characteristics of. So in terms of viruses, some examples include HIV and TMV, tobacco mosaic virus. You'll need to know about them specifically, but measles and flu are also examples. You've got salmonella, gonorrhea, which are caused by bacteria. Fungi is thrush and rose black spot, and malaria is caused by a protist. And we'll look at that in a bit more detail later on. So how can we spread these pathogens? Well, there's 4 pictures. At this point, I'd like you to pause the video and then have a think of what those 4 pictures might represent in terms of methods of transfer. When you've tried your best, you can restart the video. So pause the video now. So there are 4 main ways of transferring bacteria, viruses, protists, or fungi. And they are by direct contact, through water, through the air, often seen as water droplets, and through what we call a vector. And remember, if you go back to the earlier part of the presentation, you need to know what the definition of a vector is. What you've got in front of you is an example of modern medicine versus early medicine. And all I want you to do is spend a minute, just have a look at some of the examples of how things have changed, and in particular, how things have changed to reduce the spread of pathogens. So pause the video, take a minute, have a quick look, and see what you can see. Well, some of the obvious differences are the number of people watching. First off, everybody in the modern surgery is there for a particular reason. No one's there just to watch. Whereas in the other picture, everyone's there to watch. They've also got masks. Most of what's going on is now covered. We work in a clean room, whereas the other room you can see there's wood and all sorts of stuff there. It's not particularly hygienic. In the modern surgery, you'll obviously have much more cleaning. You will have much more wiping of surfaces, even simple things like hand washing. You would have had in early surgeries doctors going from one surgery, possibly working on a corpse, and then going to another surgery without washing their hands at all. So there's an awful big difference, and because of that, we have a much higher survival rate in surgery these days. There are two people you need to be aware of in terms of the immune system and pathogens. Ignaz Semmelweis is basically a doctor who brought in the concept that whenever a doctor is finished with a patient, and the patients they worked with were newborn mothers, or they were finished with some kind of dissection or some kind of autopsy, he expected all his doctors to wash their hands and make sure their hands were clean before moving on to the next mother. And basically what was happening was previously you had an awful lot of pathogens being transferred between the mothers, and an awful lot of newborn babies and mothers were dying. And when Ignaz brought in the hand washing rules, it had a huge impact in the survival rates. In his hospital, people were much more likely to survive childbirth. Florence Nightingale did a very similar thing during the Crimean War. She was based in basically a field hospital, and again, a lot of what she did was very simple things like washing hands, washing down tables between patients and things like that. And again, it greatly reduced the spread of pathogens, which meant a lot less people were dying from disease and infections. So how do we stop transmission? We've said the four methods. There are a number of examples of things that we can do that will stop the transmission of pathogens. At this point, pause the video, try and think up as many examples as you can for each of the different methods of transfer, and then we'll go through the examples you need to know for the exam. Pause the video now. So in terms of direct contact, the main ones are basically hand washing, keeping your distance, vaccinations, and we do have to talk about barrier contraception, because direct contact would of course talk about sexual intercourse. So any type of condom is a barrier contraceptive. It doesn't matter which type of condom you refer to, male, female, or otherwise, they are all the same thing. They're all an example of barrier method. The only other method, of course, is keeping your distance, which would mean abstinence or not having sex. In terms of other direct contact diseases, keeping your distance, for example, keeping a 2-meter rule and hand washing and so forth, will help stop the transfer by direct contact. For those in water, what you basically have to do is sterilize the water. And there's a number of ways we can do that. We can add chlorine, which is a chemical that they'll add into swimming pools and so forth, and it'll basically kill any of the pathogens inside the water. We can also use UV light, and UV light can be used to sterilize water. For air traveling, we can talk about wearing a mask, covering our mouths, coughing and sneezing. We should even go a little step further. In real life, it's not just covering your mouth when coughing or sneezing. You need to wash your hands afterwards, because anything that can spread by air often can spread by direct contact as well. And again, vaccinations are a huge part of prevention of transmission. And the last one in terms of vectors is if the vector is an insect, we can use pesticides to kill it. And basically, if we kill whatever's carrying the pathogen, we kill the pathogen and stop it being able to transfer. And in particular, often we talk about the vector being mosquitoes, and we talk about putting nets or mosquito nets over beds so that when people are sleeping, they don't get bitten. So just to finish off, I want you to think back to the questions you should have memorized from the start and see, can you do those questions from memory? So have you gone through, taken time to memorize? If you haven't, pause the video now, learn it off by heart, and then come back and have another go.